Okay, so for structured question 2 of the photosynthesis tutorial, the location where RUBP and GP are produced would be the stroma, because these two are intermediates of the Kelvin cycle which happens in the stroma. Right, then for part 2, looking at the changes in the concentration of RUBP and GP, right? Here, right, you need to note one concept when we come to looking at a cycle, like a Kelvin cycle, right? For any one of the intermediate, for example, our RUBP, right, its concentration is always a net between the formation and its usage. Because at any one time, right, let's say for RUBP, while it is being used in carbon fixation, it is also being regenerated at the same time. Right? So when we look at the initial part, right, of the graph, when RUBP concentration is constant, it is not like nothing's happening, right? RUBP is constant because the rate of usage and the rate of formation are the same, right? So there's no net change. So when we want to explain for the increase in RUBP concentration, right, then you need to explain in terms of both the formation and the usage. Okay, so I'll show you my answer and you see how that is done and you see what I mean. So for the 4 marks, right, the first point, go to the description. Of course, you must quote figures, right, quote values. So you do the description of the changes for both RUBP and GP. After that, I give the basis, the main theoretical basis for the changes. So in this case, because it's due to the decrease in the carbon dioxide concentration, right? So that affects the carbon fixation in Kelvin cycle. Okay. Then we look at RUBP itself. Okay, so because there's less carbon fixation, so the usage, right, of RUBP is reduced, right? Less of it is used to combine with the carbon dioxide. Okay, but at the same time, right, RUBP is still regenerated from whatever TP that is already existing in the stroma. Right, so we have less usage, but the regeneration still continue. So that's why for that short duration of time, the concentration of RUBP increase. Okay. As for GP, right, because there's less carbon fixation, right, so there's less of the unstable 6-carbon compound formed, and therefore less GP is formed. Right, and whatever GP that's already existing is still being reduced to TP, right? So there's a net decrease in the concentration of GP. Okay, so if you look at this explanation, right, then I hope you understand what I meant by saying that when you explain for the change in the concentration of the different intermediates, you must look at both usage and formation. So again, for RUBP, less usage because of the decrease in carbon dioxide concentration, while there is still the same formation, right? So less usage, same formation, overall net increase. For GP, there's less formation, but same usage, right? So there's overall net decrease. Okay, so this is how you explain for the changes in the concentrations of the intermediates, because it's a cycle, right? So you must look at both the formation and usage, which happen at the same time, right, to affect the overall concentration of the intermediates at any one time. Okay. All right, then move on, to part B, right? So for part B, there is a decrease in the concentration of GP, and that is linked to a decrease in harvest. Right, so somehow your suggestion or your explanation that you suggest here must link to the decrease in harvest, right, as mentioned in the question. Okay, so you must talk about the growth of the plants being affected, because that's what harvest is all about, right? Either your plants are smaller in size, right, or there are fewer of the plants. right? So somehow you must link it to the effect, or rather you must link it to how the growth of the plants is affected. Okay, so obviously with a decrease in GP, there's a decrease in TP as well, because there's less reduction from GP. Okay, and with less TP, then you know that TP forms the glucose phosphate, right? So there's less glucose formed, and the plants being producers, right, they make all their biomolecules actually from this glucose. So this glucose obviously can be used to form carbohydrates like cellulose, which if there is not enough cellulose being formed, then obviously that affects the growth of the plants, because when the plants grow bigger, right, there should be more cell wall, and therefore more cellulose is needed. Okay, and the glucose again can be used to make other biomolecules as well, like proteins and even fats, lipids, right, all of which are required for growth. And also, you may also mention how less glucose, right, also means that there's less energy from respiration, which is needed for growth again, growth and reproduction, right? So you can also explain via the energy from respiration route, okay, if you did not talk about the production of biomolecules, right, needed for growth. Okay, so any reason, right, logical reasons that link to how growth of the plants is affected, basically. Okay, then move on to part C, right? So for part C, right, the question mentioned the illumination of chloroplasts, right? So that would be the starting point, and that would eventually lead to the high pH in the stroma. So high pH in the stroma means that the concentration of H+ in the stroma is low, right? High pH means low concentration of H+ in the stroma. Okay, so you got to explain by looking at how the process happened, starting from the illumination of the chloroplast, meaning when light shines onto the chloroplast, right? So starting from that, you got to mention the absorption of light by the photosynthetic pigments, right? The accessory pigments and chlorophyll, and how the light energy that is absorbed is passed to the reaction center, right, with the chlorophyll A molecules, which causes photoactivation, right, the excitement of electrons. After that, right, then the process continues with the excited electrons being passed to the ETC, and the electron carriers in the ETC, right, using the energy from the electrons to pump H+ from the stroma into the thylakoid space. Okay, so because H+ are pumped into the thylakoid space, right, so there would be less of them, right, in the stroma, and therefore giving the high pH in the stroma. Okay, so starting from when light, reaches the chloroplast, all the way to when the H+ are pumped, right, away from the stroma, right, into the thylakoid space. Okay, so this question, right, basically is testing you on your knowledge of the process, right, of photoactivation, followed by part of photophosphorylation with the ETC pumping the H+. Because we do not need to go on to talk about ATP production, because that's not part of the question. Right, so we can end with the H+ being pumped away from the stroma, right? So therefore giving the high pH. Okay, then for part D, right, you will need to look for structures that are specific for bacteria. So once you see these structures, you know this must be a bacteria, and these structures, of course, must also be found in the chloroplast. That's how we can conclude, right, that chloroplast used to be bacteria. All right, so the structures would be circular DNA, which are specific characteristics of the bacteria, as well as the 70S ribosomes. Okay, which of course both are found in chloroplast. and mitochondria as well, right? So the same endosym biotic theory, right, applies for both organelles. Okay, and for the last part, right, the question is kind of similar to our A part 2, right? There is still changes again to the concentration of GP and RUBP, but this time round, instead of a decrease in carbon dioxide concentration, it is a decrease in light intensity, right? No more light. Okay, so the way to explain would still be similar. You need to look at both the formation and the usage, all right? So I hope you have learned how to do it from A part 2, right? So I suggest that you maybe can pause the video now and try to update your answer before you check mine, right? So that you give yourself a chance, right, to learn and check your learning instead of just copying, because such questions, right, that you see twice in this one tutorial question, these questions are quite common, right, in the A levels, in school exams, other school exams as well, right? So you should know, or you need to know, right, how to explain such questions very well. So I would encourage you to pause the video and we'll try doing it yourself first. Okay. All right. So now I'll just show you my answer for you to check. Okay, so the structure of the answer is quite similar to A part 2. I will still have 4 points, even though this is a 3-mark question. Right, so the first point will still be the description, but no QV here because no numbers are given in the graph. Okay, then the basis of the change. So in this case, because it's a lack of light, right? So we have photophosphorylation that will be affected, and therefore no products, right, from photophosphorylation, namely the ATP and reduced NADP, right? So because of that, right, for our GP, right, carbon fixations still occur because there's no change to carbon dioxide concentration, right? So GP is still formed. However, because of the lack of ATP and reduced NADP, GP is not reduced to TP, right? So we have formation, but no usage, right? Formation, no usage. Therefore, there's a net increase in GP. As for RUBP, right, it continue to be used because again, there's still carbon fixation. However, because of the lack of ATP, right, any existing TP cannot be regenerated into RUBP. And of course, over time, there will be less TP even, right? Because of the as explained in point 3, right? The lack of reduction. Okay, so overall, there's less regeneration of TP to RUBP while it is still being used. Right? So no formation, but got usage. So therefore, there's a net overall decrease in RUBP concentration. All right. So again, another example to show how to explain with both formation and usage. Okay, so I hope you really get to learn how to do this, right? Quite important. Okay. Okay, so that will be question 2. Yeah.